Good afternoon, everybody. I'll be talking about antimicrobial resistance in the environment. And maybe to begin with, I'll just go back to 2015. That's when WHO sounded the alarm bells for antimicrobial resistance and the growing threat. So subsequently, WHO published the priority hit lists for the pathogens. The two top ones, critical and the high priority lists, are shown here. And you can see that these pathogens that are resistant to the last resort antibiotics, such as the carbapenems and the third generation cephalosporins, are high on the list, as well as many others. So it's a very difficult problem to solve. And as some of the earlier speakers have alluded to, what's important now is to try and look at it from a holistic point of view and to apply this One Health approach. Right? So it's a multifaceted problem. So we have to approach it from the three sectors: the animal health sector, the human health sector, and the environmental health sector. And that's where I come in. I come in and I bring in the perspective from the environment. So most of you already know that antimicrobial resistance comes about from the inappropriate and overuse of antibiotics, not just in the human health sector, but also in the agriculture and veterinary medicine. There are multiple sources, and most of these originate from land-based activities. As you can see, the diagram is very complex, but essentially, we have fecal contamination, we have fecal matter, as my previous speaker alluded to, that is being applied to agriculture, and this contamination then fuels the antimicrobial resistance. So in the environment, we have three major compartments to deal with: soil, water, and air. And these naturally act as hotspots for the circulation of antibiotic-resistant bacteria, as well as antibiotic-resistant genes. And we are, of course, concerned about the exchange of these genes and and how it might lead to multiple drug resistance and ultimately implications on human health. So from the environment perspective, I think the two major sources are probably going to come from sewage. And water reclamation plants, or actually their wastewater treatment plants, is one major source, and the other is storm runoff. Now, if we look at the wastewater treatment plants, in Singapore we call them water reclamation plants, but they're essentially the same thing. The sources of antimicrobial resistance comes from the domestic sewage or community sewage, as well as from industry, hospitals, and environments where feces are discharged, like maybe through animal or wildlife parks, animal farms, where the sewage is collected and then discharged into the reclamation plants. From the other perspective, if we look at the non-point sources, which are more diffuse, we have stormwater runoff that collects fecal matter, antimicrobial resistance determinants, such as your antimicrobials, coming from animal farms, agriculture, urban runoff, as well as aquaculture. And ultimately, all these different sources can feed into the aquatic environment, which is the focus of my talk. So my research area is looking at antimicrobial resistance in aquatic environments. Now, along with the sewage or the wastewater, we know that antibiotics are discharged, and we know also that the low concentrations of antibiotics that don't kill off the bacteria can ultimately lead to the development of antibiotic resistance. But in the wastewater, there are other chemicals that may be present. For example, other biocides, disinfectants, detergents, and even heavy metals. And these other chemicals that are present in wastewater, together with the antibiotics, actually enhance or increase the antibiotic resistance of the bacteria. And so this is one of the things that we are also interested in, and we're trying to see how these correlate to the growth of antimicrobial resistance in the environment. Ultimately, the bacteria that acquire the resistance can transfer the resistance through horizontal gene transfer to other species of bacteria, or perhaps to the next generation through their chromosome. So these are some of the issues that we are looking at: how, for example, the chemical pollutants, the multi components of chemicals can affect the microbial community, and then that in turn, how does that affect antimicrobial resistance and its dissemination in the environment? Maybe I'll just give you some background, some studies. This is a study that was done on urban sewage and using metagenomics to see the different microbial or antimicrobial antimicrobial resistance across different countries. On the, oh, let me see, I can't, yeah, here's my mouse. Not sure if you can see it, not really. If you look at the graph on the left, that shows the total AMR for the different continents. So there's Africa leading the pack with South America, followed by Asia. And this was a study that comprised about 60 different countries. And if you look at the right-hand panel, you'll see that the resistance profile actually shows some patterns. Macrolides, which are in brown, tend to be the dominant resistance, followed by, oh, well, not what, as well as tetracycline is also, tetracycline resistance is important, as well as aminoglycoside and beta-lactam resistance. What is interesting, though, is that if you look at Europe and North America and Middle East, they actually show similar patterns. And these are different from, this pattern is different from Africa and Asia, which show similar patterns. If we look at the relative abundance of the top 15 most common ARGs in the bottom panel, we also see similar patterns where the profile for Europe and North America is similar and contrasts sharply with those of Africa and Asia. So ultimately what we are trying to understand then is what is the link, what are the links between the aquatic environment and human health? You know, do we see any patterns of resistance? And do we see any mechanisms of transfer? And these are some of the questions that we wish to address. What are the significant antibiotic resistant bacteria, antibiotic resistant genes, and antimicrobials of concern? What are the major sources in the aquatic environment? And what are the impacts on human, animal, and aquaculture health ultimately? And with that understanding, how can we better manage AMR in the environment? So these are broad generic questions, and we're interested in these questions in the context of Singapore. And Singapore, as you know, is a very densely populated country. We have 5.6 million people packed into about 700 square kilometers. And so there is this very close proximity between people and the environment. In Singapore, we have a very well-developed or very well-managed urban water cycle. From this diagram, you can see that we collect our rainwater, and this is subsequently treated to a high degree, and then is sent out as potable water for domestic use and industries. The used water, which is the wastewater, gets collected, and we treat that used water to a high degree, and the effluent is discharged into the sea, but a large proportion of it now is treated even further. It's what we call reclaimed and converted to new water, which is essentially as good as potable drinking water, but we actually put it back into the reservoirs, which as a to supplement the raw water sources, and then it goes through the conventional water treatment. So that's how Singapore manages its water cycle in a nutshell. And from the AMR perspective, we were interested to find out what are the baseline levels of antimicrobial resistance in the surface waters, that's the freshwater, the seawater environments, as well as the used water and the wastewater treatment plants and the water supply treatment plants. So our research objectives were to, first of all, identify and quantify the kinds of ARB, ARGs, and antibiotics in different environmental compartments, and then to build baseline resistomes databases for risk assessment. And this would mean the development of models to help us better predict the AMR in the environment, and also to identify source-associated AMR biomarkers. So we set out to develop the methods to do this, basically to look at ARB, ARGs, and antibiotics, and we apply them to different environmental compartments, and then we conduct our risk assessment. Well, actually, we develop the models to do the risk assessment. So that's the overall framework of our research, and I'll go into the specifics shortly. So for the methods, our overall analytical approach is to combine not just the microbiological analysis, but also the chemical analysis. So here we have culture-based methods looking at the growth of antibiotic-resistant bacteria on selective media. We also look at the diversity of the bacteria community, the resistant community. We look at the resistome, the antibiotic-resistant genes. We also conduct antibiotic susceptibility testing using the VITEC2 instrument. And we also do chemical analysis using LC-MS as well as QTOF to look at the using a non-targeted approach. So we try to look at it in as holistic a manner as possible, going from the antibiotic bacteria, resistant genes, as well as the antibiotics and antimicrobials. So for the microbial analysis, we focus on 5 bacteria, Klebsiella pneumoniae, Pseudomonas aeruginosa, E. coli, Enterococcus, and Vibrio for the seawater samples. And we culture these on the specific media on plates which have been supplemented with specific antibiotics, as well as plates that have not been supplemented. And then we pick the isolates, and then we do subsequent testing for their identity, right, using phylogenetic sequencing, 16S, and then also susceptibility testing with VITEC2, and also a screen for selected ARGs. We also take the whole water sample and extract the DNA to also do some metagenomics and resistome analysis. And ultimately, the data feeds into the development of our risk assessment models. So this is a table showing our target antibiotic-resistant genes, which cut across the various antibiotic classes listed here. We focus more on the beta-lactam antibiotics because they're the last resort antibiotics. And we also use high throughput qPCR for some of our samples, where we can analyze up to 250 genes at one shot. For the antibiotics and antimicrobials, we have about 26 chemicals that we analyze. More recently, we have added into our list the antivirals and disinfectants, which are used as a result of COVID. And we've also started using a non-targeted approach using QTOF LCMS. In terms of the environmental samples that we've been looking at, you can see here that we have covered quite a diverse range of environments, including catchments and reservoirs, seawater, aquaculture farms, hospital wastewater, domestic wastewater, animal sources, including a pet center, a huge pet center, whoops, and also our drinking water treatment plants, as well as our wastewater treatment plants. So here is a summary of 3 of the opportunistic pathogens that we've been monitoring in the various wastewater and environmental waters. We have E. coli in the first panel, Enterococcus in the second panel, and Pseudomonas aeruginosa in the third. Now, it's a very busy graph, but if you look at the first panel, which is the E. coli, this, by the way, is just the susceptible bacteria concentration. So these are not the antibiotic resistant ones. This is just the total average concentrations that we find. As expected, the concentrations in wastewater are typically about 10 to the power or 3 to 5 orders of magnitude larger than what we see in the surface waters. Okay, so for the wastewaters, we have hospital wastewater, wastewater treatment plant influent. We have actually wastewater collected from manholes from HDB housing estates, which are very highly densely populated. We have workers' dormitory as well, and manhole sewage collected from a large pet center which houses several hundred animals, pet animals. And then we also collected water from marine environment, aquaculture farm, and freshwater. So the general trends across the three bacteria are similar, right, with much higher concentrations as expected in the wastewaters. In the surface waters, on the, what we see here, the dashed red line represents the WHO guidelines for recreational waters. So for the case of fresh waters, these meet the guidelines, but for the marine waters, they don't quite meet the guidelines. Now, if we move to the antibiotic-resistant bacteria, this panel of graphs shows the average concentrations of E. coli with resistance to 5 antibiotics. So the 5 antibiotics are listed here: amikacin, ciprofloxacin, cotrimoxazole, ESBL, and KPC. The first panel represents the no antibiotic plate. So that's the total concentrations that are not resistant. So from here, for at least E. coli, we can see high levels of resistance to ciprofloxacin, cotrimoxazole, ESBL, and slightly lower levels for KPC. And in the case of the freshwater environments and the marine environments, the surface waters, right, we see very low levels of amikacin resistance and also KPC, low resistance in KPC for aquaculture. If we focus now on the ARB in the community sewage, we took samples from an HDB or two HDB housing estates, workers' dormitory, as well as the pet center with hundreds of animals. And from this, if we just look at the concentrations of the four pathogens, we see that Klebsiella pneumoniae had the highest concentrations, followed by E. coli and Enterococcus, and Pseudomonas aeruginosa had the least concentration. But when we look at the percentage of multi-drug resistance, both P. aeruginosa and Enterococcus actually had almost 100% multiple drug resistance. And if we look at specifically the pet center, I think this is a bit off, but the pet center actually showed the highest percentage of multiple drug resistance for the samples that we took from these different wastewater types. We also looked at the metagenome for these different wastewaters. So on top of the 3 wastewater types that we looked at earlier, we also included wastewater from the wastewater treatment plant as well as from hospital sewage. And in the panel, in the graph on the right, you'll see that red represents the wastewater treatment plant, orange the hospital sewage, and blue is the community sewage. So here you can see that the hospital sewage as well as the wastewater treatment plant influent had the highest relative abundance of antibiotic resistant genes. And these were associated also with biocide and metal resistance genes that were also present. Right, so that's seen in the lower graph as well as in this is the genes are seen here. On the graph on the right, we saw there was clustering of the antibiotic resistant genes for the domestic sewage, meaning the residential sewage and the workers' dormitory, and this was separately clustered from the pet center, which was in green, and the hospital and the wastewater treatment plant influent samples. So next we tried to see whether there were any markers, antibiotic resistant genes that was more typical of specific sewage types. And so here we actually saw that for the pet center, the animal-based wastewater, the markers generally showed resistance to the first-line antibiotics, tetracyclines, sulfonamides, and as well as aminoglycoside. Looking at the hospital, though, the hospital had a very distinct profile. The antibiotic resistance was mainly for the beta-lactams as well as the aminoglycosides. And when we looked at the residential wastewater, domestic sewage as well as what was coming into the wastewater treatment plant, they had similar profiles, mainly with multi-drug resistance. So we did see these different patterns for the different sewage types. And when we looked at the proportion of ARGs carried on chromosomes and plasmids, right, we actually saw that most of the ARGs were associated with plasmids. And most of these, in turn, were actually from the hospital samples, as well as from the PET center. And so we found more plasmid-associated ARGs with these samples. And similarly, we saw these ARGs co-occurring with mobile genetic elements and integrative conjugative elements as well. We also analyzed antimicrobials in the sewage types. We have a list of about 26 antimicrobials shown in the table on the right, and the graph on the left show the concentrations of the different antimicrobials, with ciprofloxacin generally leading the pack, followed by erythromycin. So the concentrations ranged from almost non-detectable to 44,000 nanograms per liter. We basically tested all 26 antimicrobials, but only 16 were detected in the community sewage, and 13 of these were across all the different sewage types. Beta-lactams were not present except for amoxicillin. And for comparison, when we measured the concentrations in the coastal or marine samples, right, they were several orders of magnitude lower, right, maybe only up to about 200 nanograms per liter. So here's the data that we have from the marine environments as well as the aquaculture sites. We actually sampled from, this is a preliminary data, we had 2 beaches and 2 aquaculture farms that we sampled at. And the mean concentrations, or the median pathogen concentrations, as you can see, was much lower, around 10 to 1,000 CFU per 100 mil. This is the antibiotic resistant profile for E. coli. And here we saw that for one of the beach samples, it showed particularly high multi-drug resistance to 12 of the 16 tested antibiotics. Right? So these are the different antibiotics on the x-axis. And what we see is, in fact, penicillins resistant was pretty dominant for all the water types, but carbapenem resistance was still quite low, as you can see here. In fact, was generally less than 8%. Looking at Enterococcus, the profile was quite different. Here, one of the fish farms had very high multiple drug resistance to 11 out of 15 tested antibiotics. And here, the MLSB resistance tended to be much higher for Enterococcus. So in this table, I would like to compare the findings that we have for our local waters with other countries. So the column in red shows the data that we collected in Singapore. And if we compare this, say, the penicillins across the other countries, this is data that we managed to compile from the literature. We are basically, compared to India, right, we still have slightly lower resistance, but comparable to Canada, at least for ampicillin. For the aminoglycosides, we have roughly the similar resistance. Fluoroquinolones, we are doing better in terms of the resistance, lower resistance compared to India, and similarly for the cephalosporins. So this is just to compare with what other people have been finding in their coastal waters. More recently, we've been interested in the marine environment, marine plastics. And here we took samples from various mangroves and beaches around Singapore. We collected not just the water samples as well as the sediment samples for antibiotic resistant determinants, but we also looked at the plastics because, as you know, plastics can potentially be a vector for biofilms and antimicrobial resistance. So here we looked at the different types of plastics that we collected and we sized them as well. And we tried to look for the bacteria and the resistant genes that were growing on them. So this is a cluster analysis of the different plastic types, which is a very messy diagram, but essentially what we could do is we could differentiate between the resistome found in the water column, and we saw that this was quite different from what we saw in the sand as well as in the plastics. The overall ARG abundance was also much higher for the plastic associated resistant genes. So with the data that we've collected from the different environments, we use this data to try and construct models that can help us to better predict the risk from the different AMR determinants that we have measured. So here we've taken both antibiotic-resistant bacteria as well as antibiotic-resistant genes and tried to construct a model to give us a relative risk assessment of the AMR from different environmental waters. In order to classify the ARB, we make use of the DALI's database, which prioritizes different kinds of antibiotic-resistant bacteria. So the WHO has put out this list, and we make use of this list and assign risk coefficients to the different antibiotic-resistant bacteria. Similarly, for the different antibiotic-resistant genes, depending on what they are, like the blob have a higher priority or compared to others, we also associate with them a certain risk coefficient linked to their significance and importance. So from our environmental data, where we measure the concentrations, we normalize this, and then we multiply it by the risk coefficient to get some sense of how important the different ARBs and ARGs are relative to each other. And then we combine this data to get a score, which gives us the relative risk of the AMR. And so this is what we've been able to use our data to then develop weighted AMR risk scores for our different samples. So for example, here we see that samples in December during the northeast monsoon tended to have a higher AMR risk compared to other times. And in the right-hand panel, we can see how different land use can result in different risk levels as well for different activity types. So here, for example, agriculture, where water is going through tributaries in an agricultural catchment, would result in a higher risk compared to the other zones. Finally, more recently, we've tried to combine or develop a new model which tries to combine QMRA with the WHO DALI's report for a novel way of looking at risk assessment for AMR in the environment. Normally, when we look at pathogens in the environment, we use QMRA to estimate the probability of infection. But using just QMRA does not tell us how severe the problem is due to ARB. So what we've done is we've taken the data from our environment, where we have both susceptible as well as antibiotic resistant, say E. coli here. We've calculated the probability of infection using QMRA, and then we've combined this with the DALI approach, where DALI accounts for the disability adjusted life years. And in the DALI approach, you have health outcome parameters that are included. For example, fatality proportion, length of stay in the hospital, and different significance or health severity of the illness. And so we can combine all this and get a DALI baseline and a DALI value related to antibiotic resistance. And this difference then gives us the change in DALI, which is the additional health burden brought on by the antibiotic resistant bacteria. So this is a relatively new approach, and it's something we've just started working on, and we're hopefully, with the data that's coming out in the coming months, we will be able to calibrate this further. And so with that, we know that this problem of antimicrobial resistance is a very difficult one, but hopefully, as we look at this from a One Health perspective, and me coming in from the environment, right, we can try to better understand the sources of AMR, the mechanisms involved, the occurrence and the prevalence of the different AMR factors, right, we can help to fit in this jigsaw puzzle and solve this problem together. So with that, thank you very much.